Welcome back. This is day 3 of asexual reproduction. Today's aim is how does mitosis form identical daughter cells? But first, you guys have got a do now. So go ahead and pause this video and tell me, why must every cell produced from asexual reproduction receive a complete copy of DNA? Okay, and press play when you're ready to review it. All right, so remember that DNA is basically a set of genetic instructions. Okay, they tell your cells how to function. We described it as kind of like the instruction manual. Whenever you get a set of new IKEA furniture, right? If you only get half of those instructions, are you going to be able to build that entire dresser? No, you need the full set of instructions in order to finish building it. The same thing happens with our DNA. If our cells don't have that full set of DNA, they're not going to work correctly. And since with asexual reproduction, we only have one parent, that full set of DNA has to come from that one parent in order for that cell to work properly. So with asexual reproduction, we produce genetically identical cells, and we have to have that identical DNA in order for those cells to function. All right. Now, today's aim is, I've already said it, but how does mitosis form identical daughter cells? But what exactly is mitosis? Yesterday, we learned that mitosis is a type of cell division where one cell divides to create two genetically identical cells. And this is something that's happening in your body right now. This is how we make new body cells. Another word for body cells that you'll hear pretty frequently is somatic cells. That word somatic is just another way of saying body. So somatic cells or body cells are things like your skin cells, your muscle cells, your nerve cells. Those are all examples of somatic cells. So in order for this to work properly, the nucleus has to divide. It has to duplicate and divide to actually form two nuclei, so that when that cell eventually splits, each resulting cell, each daughter cell, is left with an identical copy of DNA. So again, we're still making genetically identical cells. Okay. Here's another picture of what that looks like, right? We start with the parent cell. That DNA is going to replicate or duplicate. So then eventually when the cells split, each daughter cell is left with an identical copy of DNA that is absolutely the same as that parent cell it came from. Okay, but what exactly is cell division? What is this process of cell division used for? Why do we need it? Let's find out. So we're going to read through this paragraph. There are a few blanks that you want to fill in. If you want to pause this video and take a guess and see what you already know, be my guest, right? Fill in those missing blanks and then press play when you're ready to review it with me. Okay, so cell division, what is it? This is the orderly separation of one cell into two. Before a cell divides, the genetic information in the DNA of the cells is duplicated or replicated exactly. That means we now have two complete copies of it. So then during cell division, one copy of this information is distributed to each cell. As a result, each new cell has all of the information it needs to function properly. One-celled organisms make use of cell division for asexual reproduction, but multicellular organisms mainly use cell division for three things: growth, cell replacement, and repair. This process, by which a cell's genetic material divides, creating two complete sets of the cell's genetic material, is known as mitosis. So mitosis produces two cells that each have a full set of identical genes and chromosomes, unless something called a mutation occurs along the way. Now, we will be talking more about mutations later on in this unit, as well as when we get to our genetics unit. But for right now, I just want you guys to know that mitosis in multicellular organisms like us is used for three things: growth, cell replacement, and repair. Those three concepts will come up again, so make sure you're comfortable with that. So an example of this, right? Whenever you fall and scrape your knee, that cut doesn't stay forever. Eventually, your skin is going to grow back. And that is a result of mitosis, your cells dividing and creating new cells to repair that wound. Okay. Now, we've talked about how the DNA replicates inside the nucleus, but what exactly does that look like? Well, let's find out. So we need to recall that DNA replication makes an identical copy of all genetic information in the molecule. This replica carries the instructions for the same proteins as the original strand. When the DNA replicates, it's actually turning a single-celled chromosome into a double-stranded one. This double-stranded chromosome then has a duplicate set of instructions to pass on to each of two cells. So remember, the DNA is like that IKEA instructions manual. You have to have the complete, full instruction manuals in order to build all of the right materials. The same thing happens in your cells. Each cell has to have an identical copy in order for those cells to make the right proteins. So in order for that to happen, these single-stranded chromosomes have to duplicate or basically multiply themselves by 2 and form what we call a double-stranded chromosome. Okay, and this is when we see this word chromatids coming about. So we've heard this word before, sister chromatids. And remember we said that chromatids are basically one half of this double-stranded chromosome. They each carry the same genetic information. So it's basically this times 2, right? We now have sister chromatids that have the same exact genetic information inside of them. But unfortunately, these sisters don't get to stay together forever. They are going to be separated when this cell eventually divides. And so what ends up happening is one chromatid is pulled into one cell and the other chromatid is pulled into the other, so that eventually those two cells have the same genetic content as this single-stranded chromosome that we started with. Okay, here's another picture of what happens specifically in that mitotic division. So during the process of mitotic cell division, the double-stranded chromosomes that are visible during mitosis split into 2 identical strands and move apart to opposite ends of the cell. So in this image, you can see a chromosome during mitosis. When the cells divide, each double-stranded chromosome separates into 2 identical single strands. I'm going to highlight that. Hold on. Okay. This process ends when the cytoplasm divides, resulting in 2 smaller but genetically identical cells. So this is what we just saw, right? We start with one cell. The chromosomes are going to duplicate, so they double. We now have a double-stranded chromosome still inside that single cell. Okay, but remember when we have that double-stranded chromosome, we now have 2 sister chromatids that are each carrying identical sets of the genetic information. But unfortunately, we have to separate those sisters at birth because they're needed elsewhere. Because when that cell divides into 2, one sister chromatid moves into one cell and the other sister moves into the other. So now we have identical sets of that genetic information in each of our resulting daughter cells. Okay, so again, the cells that I've starred are identical. We have identical copies, genetically identical copies of those cells. Okay. Now, it's not quite so simple as that, though. It doesn't all just happen automatically at once. There's actually a bunch of different steps of mitosis that have to work just right in order for this process to work correctly. If even one step messes up, we can run into some issues. So we're going to quickly run over the different steps of mitosis that you'll see in the diagram on your paper. So this very first step of mitosis is something called interphase. And during interphase, this is basically just preparation for the road ahead. So nothing too crazy happens yet. Why is come on, PowerPoint, there it goes. During interphase, the cell is just getting ready to divide. So during this stage, the cell copies its DNA and prepares for division. And this is actually 80% of mitosis. So most of mitosis is spent in interphase, just getting ready for the journey ahead, making sure everything is copied correctly and ready to go. And then as soon as interphase is over, we move into something called prophase. And this is where the chromosomes are going to duplicate and condense. So this is where those chromosomes actually shift into double-stranded chromosomes. We have double the genetic information now. And when we say condense, it basically means they brace for impact. They kind of tighten up and get ready to be moved around a lot because there is going to be a lot of movement in this next stage. So the next stage is pretty easy to remember because conveniently, metaphase starts with the same letter as the middle of the cell. So in metaphase, those condensed chromosomes are going to line up around the middle of the cell. You can see here that they've all moved into the middle in metaphase. And anaphase is actually another pretty easy one to remember because anaphase is when those chromosomes separate and chromatids, those sisters, are pulled apart to opposite sides of the cell. So remember metaphase for middle and anaphase for apart. This is when those sisters are separated and pulled to opposite ends of the cell, because the last step of this, what needs to happen is those cells have to split. We have to get those identical genetic copies into different cells. And that happens in the phase telophase and cytokinesis. Okay, and this is when the cytoplasm divides and splits into those two genetically identical cells. Right? And that is how we turn one parent cell into two daughter cells inside our body. The last thing we're going to go over today is cloning. Now, we briefly talked about cloning today in class, but this is another example of asexual reproduction. It is reproduction that just takes one parent to make genetically identical offspring. And my favorite example of cloning is Dolly the sheep. She was named after Dolly Parton, the country music singer. And what happened, this was in the 90s. It was actually one of the first successful attempts of cloning an animal from an adult body cell. So what happened was they took a donor sheep, okay, and this is the sheep that was going to be cloned. They took one cell from that sheep's body. They took it from the udder or the mammary glands. Now you can guess why she was named after Dolly Parton. And they took the nucleus from that donor cell, okay? And remember, the nucleus is where that genetic information is stored. But they also needed a surrogate sheep in order to make this happen, because they wanted to make sure it was actually a clone of this original sheep. So they took a surrogate that looked different, and they took the egg cell from that sheep. They took the nucleus out of it, so they removed the nucleus from this surrogate, but then they actually fused the egg cell from that surrogate with the nucleus of the sheep they wanted to clone. They put those two together, they combined them. So now we've got the egg cell from the surrogate with no genetic information, and we have the nucleus from the clone sheep. So that is where all of the genetic information is from. They combined those, and eventually those cells began dividing as normal until we reached the embryo stage of development. They then placed that embryo back into the surrogate sheep until eventually my favorite, Dolly, was born, and she developed into a normal lamb, and she lived a great life. Dolly was amazing. Let me tell you guys, she became somewhat of a national celebrity. Okay, Dolly was an icon, loved by all. She was on the cover of Time magazine. She was an idol of mine, I'll be honest. I was a kid when this happened, and I was obsessed with Dolly. She was so cool because it really just showed like the possibilities of science. This is something that had never been successfully done before. And when it happened, we were like, is science fiction becoming reality? Like, what's happening? And Dolly went on to live a beautiful life. This is Dolly right here with her sheep offspring. She had several lambs, lived a pretty normal life. She actually died when she was like 6 or 7 though, because she developed a type of lung cancer. It was really sad, but her offspring lived wonderful lives as well. And guys, Dolly is so cool. I'm obsessed with her. So I'm sorry, but you need to become obsessed with Dolly as well. I hope she's doing well wherever she is. I mean, she's dead, but I hope she had a great time while she was here on Earth. And with that being said, go ahead and complete your exit ticket for the day. There are, I think, 2 questions waiting for you. Let me know if you have any questions, and have a wonderful afternoon. I'll see you guys tomorrow.